This is Chapter 4, Sensation and Perception, Part 1. Okay, so what do you think about or visualize when you hear the word sensation or perception? When presented with a high-pitched tone, a musician reported that it looks like fireworks tinged with a pink-red hue. The color feels rough and unpleasant, and it has an ugly taste rather than a briny pickle. You've seen those ugly looking pickles, but they taste great. Okay, well, pretty much this musician was describing a rare condition that's known as synesthesia. Synesthesia means mixing of senses. People with synesthesia, they routinely blend their sensory experiences. They may see temperatures and hear colors, or they might taste shapes. So, to appreciate how extraordinarily synesthesia can be, we got to first understand the basic processes of normal, non-blended sensations. So, for an example is, how do we turn light and sound waves from the environment into something our brain can comprehend? In order for us to do this, we must first have both a means of detecting stimuli as well as a means of converting them into a language that the brain can understand. How we get the outside world to be able to be inside our brains, and what our brains do with the information. This is going to be the key topic of this chapter. Now, you know the drill. This probably will be within your assignment, so I would suggest pausing it, going ahead and looking this over, and think about the true and false to this, okay? So I'm going to go ahead and move on. So let's start trying to understand sensation. Psychologists are keenly interested in our senses. That being, the sensation is the mind's window to the outside world. We're equally interested in our perceptions, how the brain is going to give meaning to the sensory information. So sensation begins with specialized receptor cells that's located within our sense organs, that being our eyes, our ears, nose, tongue, skin, as well as internal body tissues. When sense organs detect an appropriate stimulus, like light or mechanical pressure, chemical molecules, they can convert into neural impulses. And y'all remember what neural impulses are, right? Action potentials. And that's going to be transmitted to our brain. So the brain then is going to assign meaning to the sensory information. Now, that process is called perception. Okay, and remember, perception here means the process of selecting and organizing as well as being able to interpret the sensory information and putting them into meaningful patterns. All right, so this slide helps to give you kind of an idea of what we were just talking about. I was just talking about. When we talk about our sense of vision, the stimulus can be light waves, okay? And that's where the receptors are going to be taking in the information of light waves, in which it actually becomes a picture, like looking at me right now, for example, okay? The part of the brain that's responsible for this would be your visual cortex within this occipital lobe. Auditory or audition for hearing, then we get sound waves. That's how we get these waves going in, and the pressure within it is actually the brain starts to take that and interpret it, to which you start to understand what the sounds are. Okay? And just remember, the auditory cortex is located within the temporal lobe. How we smell is olfaction. Okay? And this is where the molecules will basically dissolve, and it goes within our mucous membranes within our nose, and those neurons is going to be activated, and it allows us to kind of put meaning to what we're smelling. This would be located within the temporal lobe as well as in the limbic system. Okay, You have gustation for taste. Of course, whatever we put to our mouth breaks the molecules break down, and we begin to discern and try to figure out what it is that we're tasting. Limbic system, the somatosensory system, cortex, as well as our frontal lobes are responsible for this particular area. And then, of course, our body senses. Here, you're talking about a whole different type of stimuli, anything and everything that can come in. How we might feel the touch when we're touching our hand, when the wind blows and touch your skin. There's a variety of different receptors that's being activated. Some things in the, like within the motor cortex and the parietal lobe, those are things that's impacted. Now, you don't have to worry about this, but when we talk about how our perception and our sensation is, the demonstrating to be able to movement of detectors within the cerebral cortex and the difference between the sensation and perception. Look at this for a moment. To get the best effect, please look head on, okay? Just keep staring at it. I want you to look and it'll turn clockwise as you continue to stare, even though it started counter, it's going to move and it's going to move toward the center. After about a minute, I want you to now see if you think your head is expanding. Look in the mirror. And do you think your head is going like this? Just keep staring. All right, I'm going to move on. So, how do, sensory stimuli get within our brains in the first place? Okay. Our eyes, ears, skin, and other sense organs, they all contain these special cells that's called receptors, and they receive and process sensory information from the environment. So for each sense, these specialized cells will respond to a distinct stimulus. Like when I was talking about the sound waves, or when we were talking about the molecules that helps us to detect odors. Well, during the process of transduction, the receptors are going to convert the stimulus into neural impulses, which again are action potentials, and it sends it to the brain. So, for example, in hearing, tiny receptor cells in the inner ear will convert mechanical vibrations from the sound waves into electrical chemical signals. And these signals are going to be carried by the neurons to the brain, where specific sensory receptors will detect as well as interpret the information. So, how does our brain differentiate between sensations such as sounds and smells? Well, this comes through coding, okay? This is a different physical stimuli that's going to be interpreted as distinct sensations because their neural impulses will travel by different routes as well as arrive to different parts of your brain. We also have these structures that, you know, will help to reduce the amount of sensory information and that we would take in. And without this natural filtering that we have, we would be constantly hearing our blood within our veins, rushing, as well as feel the clothes brushing against your skin. Everything would be magnified. Our brain has learned to be able to filter things that it realized that, okay, well, you don't really need to feel your skin, feel the clothing against your skin, because that's not important. So there's some level of filtering that's going to be needed in order for our brain not to be overwhelmed. All species have been able to evolve selective receptors that will suppress or amplify information to help us to survive. And in the process of sensory reduction, what we do is we filter this incoming sensations and then try to analyze the sensations that the body does register before any neural impulse is finally sent to the various parts of the brain. Humans, for example, cannot sense ultraviolet lights or microwaves. The ultrasonic sound of a dog whistle, we don't hear that, or infrared heat patterns from warm-blooded animals, as some animals can do. So I want y'all to make sure that y'all have a good understanding of this. You want to be aware about what transduction is. Again, it's converting a receptor's energy within to neural impulses or action potentials that's going to be sent to the brain. Coding then converts that sensory information and it's going to take it into different sensations and create different sensations for you. Whereas sensory reduction, again, is that filtering process, helping us to analyze, to know what's important for us and what's not important. All right, so shown here on the slide, we hear shown here on the slide, you're going to have the primary locations within the cerebral cortex, that being vision, hearing, taste, smell, somatosensation, and that'll include our touch, our pain, as well as temperature sensitivity. Regardless of where they come from, that being the eyes, ears, nose, skin, or other sense organs, neural messages are going to be from various sense organs, and it has to travel to the brain in order for us to be able to actually see or hear, etc. Interesting enough, if we could somehow rewire these connections, sending messages from the eyes to the auditory brain areas as vice versa, we would hear light and see sound. Go figure. Now, how can scientists measure the exact amount of stimulus energy it takes to trigger a conscious experience? The answers can come from the field of psychophysics, which would be the study and measurement of the link between physical characteristics of a stimuli and the sensory experience of them. Psychophysics measures our experience of sensory stimuli. Your absolute threshold will be that smallest amount of a stimulus that you can reliably detect. Think of it this way. If you got hair on your arms, which some people do, some people don't, but if you go lightly without touching, you actually still can feel your hair moves even though you're not actually touching your skin. Think of that when you're talking about the absolute threshold. It's the smallest amount of the stimulus. That light brushing would be the absolute threshold. Okay? Your difference threshold, or otherwise known as just noticeable difference, is going to be the minimum difference that you can detect. Think about when you had last had your ears checked and they went through the different decibels. Well, what they're trying to find out is that what is your absolute and what is the just noticeably different. So when you do the ear test, it's to measure those senses. An examiner might present a series of those different signals that's going to vary within the intensity. It'll ask you to report, they'll ask you to report and say, well, what can you hear? So the softest level, that started real soft. And for me, unfortunately, I didn't hear it. It would have to get louder and louder, and come to find out to detect. Of course, I've got some damage to my ears because I used to blast my headphones in my ears when I was younger. So, hint to your Mythbusters. But what we're looking for again is the absolute threshold. That taking that in, the examiner would then try to compare your threshold with other people with normal hearing to determine whether or not you would have hearing loss. All right, so this is usually always a fun topic to discuss, and unfortunately, we're not together face to face to have this discussion. However, it is in fact possible to be able to perceive something without conscious awareness. That's called subliminal perception. Okay. There's been experimental studies that's commonly used an instrument called a tachystoscope. I'm going to spell that out. That's Tachystoscope. Tachystoscope. And what it do is it flash images quickly for your conscious recognition, but slowly enough to be registered. So when you're first looking at the object that they're using, you're watching, and within it, behind what you're initially watching, there's other images that's been placed there that, to the quickness of not thinking about it, you don't see it, but your brain actually has. Okay, that's what we're talking about when we talk about subliminal persuasion, perception. So in one study, the experimenter had been able to flash one or two pictures subliminally, and those images were either a happy or angry face, and it followed by having another picture of a neutral face. They found that this subliminal presentation evoked matching unconscious facial expressions within the participants' own facial muscles. Cool, huh? Despite this, though, much other evidence has subliminal perception occurs. That does not mean that such processes is going to actually lead to subliminal persuasion. Subliminal stimuli are basically weak stimuli. At best, they have a modest, if any, effect on the consumer's behavior. So a lot of times people think, because you see the big arcs in a commercial, that's going to make you automatically run to McDonald's. Well, it might make you think you're hungry, but it doesn't necessarily mean you're going to run to McDonald's to eat. So, and that's what they're talking about when they're saying this week. Okay. Um, and by the way, it has absolutely no effect on minds of youth that listen to rock music, or people who are voting, citizens voting rights. It doesn't, behaviors, it really doesn't affect them in that way either. As for subliminal tapes that's promising to help you lose weight or relieve stress or try to help you financially save money, those blank placebo tapes, they'll appear to be just as effective as if you just tried on your own without using those tapes. Okay, and pretty much studies indicate that. There's been research in the areas. Now, when we talk about sensory adaptation and we're talking about that weakening of the response, imagine that friends have invited you to come visit their beautiful new baby. And oh, the sweet little baby. As they greet you at the door, you're overwhelmed by the odor of that diaper because their baby done let it out. They are wet. They are oof. And you look and you wonder why your friends aren't doing anything about that smell. Well, the answer is that because previously mentioned when we talked about sensory reduction as well as sensory adaptation, that constant stimulus of smell for the parents during a long length of time, the sensation fades away and their brain says, okay, well, it's not going nowhere. So therefore, I don't want to worry about it no more. Thus, they become adapted and accustomed to the smell to the point they no longer smell it. Okay? So when we're talking about in sensory adaptation, these receptors, they're tired now. Okay, and they actually start firing less frequently than they did initially when the smell first hit you. Sensory adaptation will make sense from an evolutionary perspective. So we can't afford to waste attention and time on unchanging, normally unimportant stimuli, like I mentioned earlier. When we're turning down that volume on repetitive information, the brain is going to cope with that overwhelming amount of sensory by just filtering it down. And it allows time for us to pay attention to things that actually is important in terms of change. Sometimes, however, that adaptation can be dangerous, as like when people stop paying attention to gas leak in the house. They might have smelled it initially and they got used to it. They no longer smell it, so they're no longer worried about it. And that's when problems can happen. So although some senses like our smell and our touch adapt quickly, we never really completely adapt to visual stimuli or to those extremely intense forms of stimuli, such as ammonia, or if you got a bad burn, you're going to stay alert to those, okay? From an evolutionary standpoint, these limitations on sensory adaptation, they help with survival. Okay, it helps us to remember to avoid those strong type of odors, or to stay away from heat, or go ahead, you need to take care of that burn and keep applying burn cream on it to keep you from feeling the pain. Now, if we don't adapt to pain, the question is, how do we get through it regardless? Think about athletes. How do athletes keep playing despite having painful injuries? This football season, you see people get tackled, and I mean some of them get knocked down. Okay, how do these football players get up and keep playing? In certain situations, the body's going to release natural painkillers, and these are called endorphins. Okay, which helps with our pain perception. Endorphins are the neurotransmitter that's going to act the same way as morphine would. Have you ever had morphine, or what they call Demerol? I don't even think they give that anymore. Um, but it helps to stop the pain perception by closing the gate to the pain receptors. Okay. In addition to this endorphin release, one of the most accepted explanations of pain perception would be the gate control theory. Now, this theory was first proposed by Ronald Melzack as well as Patrick Wall, and that was way back in 1965. And according to their theory, this experience of pain really depends partly on whether the neural messages gets past the gatekeeper, so to speak. And that gatekeeper is located within our spinal cord. So normally the gate would be kept shut, okay? And that's either through impulses that's coming down from the brain, or by messages that's coming from large diameter type nerve fibers that would conduct most sensory signals, like our touch or pressure. However, when the body tissue is damaged, those impulses from smaller pain fibers are going to open that gate wide open now. Okay? So, according to the gate control theory, when you're massaging an injury, you ever, oh, I got a good one. Paper cuts. They can be painful. What do you tend to do? I know when I get a paper cut, I tend to squeeze it and I keep pressing on it. Okay? When you're doing that, you're temporarily relieving the discomfort because that pressure on the large diameter neurons kind of interferes with those pain signals. So messages from your brain can also control that pain gate. And this kind of explains how the athletes as well as soldiers can keep going despite being in excruciating types of pain. When we are soothed by those endorphins or distracted by competition or fear, our experience of pain can start to greatly diminish. On the other hand, when we're anxious or we dwell on our pain, it can then intensify. So, ironically, well-meaning friends that ask about pain sufferers, or those chronic pain sufferers, about their pain can unintentionally reinforce and increase the pain. Think about a baby, not a baby, a toddler. You ever notice when a toddler falls down, if no one says anything, the toddler generally would get up and keep going. However, when mommy says, "Oh, baby, are you okay?" all of a sudden that baby starts to cry. That's what I'm talking about. Okay? Now, research has also suggested that within the pain gate, it's chemically controlled. And the neurotransmitter that's responsible for it is called the substance P. And what it does is it opens the pain gate and allows the endorphins, allows the pain to go through, and endorphins will then close it again. Okay, so when that substance P opens, you're gonna feel the pain. But when the endorphins come, it's trying to close that gate up so that you're not feeling as much pain. Okay, Let me see, is there anything else I'd like to share? Okay, there's another type of research that we could talk about, and it found that normal sensory input, when disrupted, the brain can generate pain as well as other sensations on its own. Amputees, uh, people that might have lost a limb, they sometimes continue to feel pain. Okay, and it might be an itching sensation or a tickling sensation, even though it's been a while since the limb was amputated. We call this phantom limb pain, and that's because nerve cells sends the conflicting messages to the brain. The brain going to interpret that static as pain because it arises within the area of the spinal cord that's responsible for pain signaling. So when amputees are fitted with a prosthetic, the limbs, and they start using them, phantom pain will then start to disappear. Again, think about that pressure. It's closing the gate. Now, properties of vision and hearing. There's many people mistakenly believe that what they see and hear is a copy of the outside world. Well, the fact is, vision and hearing are the result of what our brains create in response to light and sound waves. There's three physical properties of light and sound, that being wavelength, complexity, and amplitude. Help and they help, kind of determine our sensory experience of them. Both vision and hearing are going to be based on wave phenomena, that being like ocean waves. Think about standing on a pier and you see the waves at a certain distance between them. That would be the wavelength, okay? And they pass by you at intervals. Now, if you count the number of passing waves within a set amount of time, like say there's 5 waves within a minute, then you'll be able to calculate the frequency or the number of complete wavelengths that would pass a point within a given time span. The larger the wavelength means the smaller the frequency, as well as vice versa. Waves also would have characteristics of height, and that's technically would be called the amplitude. There would be some large, which would be like, think of you getting on a surfboard and you're doing you're really surfing, okay? You got a good ride going on. There might be others that's small. Finally, there'll be some waves though that have a very simple uniform shape, which would be good for surfing. Whereas there's others that's a combination of waves, of different type of wavelengths and height. And this will result in a wave that's too irregular or that would be for surfing. Hopefully that kind of helps give you a visual idea within your mind. Now, you see, I wanted to just show you as far as visually how our receptors within our eyes, how they take in the information. We'll be speaking about rods and cones and so forth. But as you see, we think everything is up this way, but in reality, me holding up my hand, you're actually seeing me this way. It's upside down. You're looking at me, but as it reflects back out, you see me right side up. But as far as when we're talking about light waves going in, it's actually being presented upside down. Now, when we're talking about vision and looking at that stimulus, we have to think about the electromagnetic spectrum. Now, this is basically, when we're talking about the full spectrum of electromagnetic waves, this contains very long wavelengths, which we can consider AC circuits as well as radio waves. That's at one end, and then relatively short cosmic type rays that will be at the other end. Also, only light waves in the middle of the electromagnetic spectrum can be seen by the human eye. So I want you to note that the longer the visible wavelengths are, the light waves that we see as red, and then those shorter ones are going to be perceived as blue. In between are the rest of the colors. So to be able to appreciate how our eyes turn these light waves into what we call vision, we have to kind of examine the various structures within our eyes as they first capture and focuses those light waves, and then how they transduce or transform, but I want you to remember transduce them into neural messages or action potentials, which will allow our brain to then process into what we consciously see. I'm going to bring these up real fast. Be sure, though, when you're studying, make sure you're looking at the step-by-step process of how this all works, okay? Because probably on the next exam, it's going to have some questions for you. When we thoroughly can understand the process, it helps to give us clues for understanding those visual imperfections or things that's abnormal. So, for an example, small abnormalities within the eye sometimes can cause images to be focused, in front of our retina, and it's considered to be nearsightedness. Also, nearsightedness is also considered to be called myopia. So you want to remember myopia, nearsightedness. Whereas farsighted, which is further away, is hyperopia. Now, with corrective lenses, or if you have the laser surgery, it can kind of correct most of the visual acuity problems that you might have. As we get older, times will change, okay? During middle age, most people's lenses tend to start losing elasticity, and that's going to lose that ability to be able to accommodate for near vision. This would be called presbyopia, and that's going to be normally treated by just getting a pair of glasses. If you kind of walk into a dark theater, for example, on a bright sunny day, the first thing that's going to happen, you're going to be like feeling you're blind. That's because of the bright light. That pigment within the rods kind of get bleached out for a moment, making them temporarily non-functional. And it'll take a second or two for the rods to kind of start functioning again, well enough for you to be able to see. This process of dark adaptation, it takes about 20 to 30 minutes. And with light adaptation, the adjustment will it'll take place when you leave the dark and start heading back into bright settings. And that takes between 7 to 10 minutes. And once that happens, that means your cones are now functioning again. Okay? Let me bring all of this up. And I want you to be able to take a pause, okay, and look at the difference between the rods and the cones. So this demonstration shows differences between the rods and cones. And what I'd like for you to do is kind of think of yourself as an experimenter. And the experimenter stands behind an object, and they're going to choose a really bright colored item, that being a pen, a pencil, a ruler, whatever's in front of you to use. I want you to then kind of fixate on a distant point. Then allow yourself to slowly move the object from your front of you to your peripheral. If this movement is slow enough, then you should be able to detect the object before you determine the color of it. And it's a little hard for you to do, so you might want to see if you can have someone do it for you. If you're with someone, have them do it and move it so that you can get the true full experience. And then one last note, please make sure to get the understanding about the retina, the rods, as well as the cones. You want to know what each responsibility is. Review that again here within the slide. And this is going to be the end of part 1. I will pick up again with part 2 shortly.